Integrated Science, Paper 2, Practical. Integrated Science, Paper 2, Practical, Grade 9. This is how we should do it when it comes to a national exam. So we have 3 tasks. Task 1, ascending-based indicator. With me, I have an indicator here by the name phenolphthalein indicator. It is labeled as solution X. Then 2, we have the cell. Something that you know about, you want to know more about cells. Good. My name is Teacher Ayub Mwiti. Let's start with the procedure. So the procedure is, you are provided with a solution labeled A, B, C, and D. You are required to carry out text using indicators labeled X. You are required to carry out text using indicator labeled solution X to determine whether those solutions provided are acid, base, or neutral. Record your observations and conclusions in the spaces provided. 1. Add about 2 to 3 drops of solution X to the test tube with the solution A and record the observations and conclusions to mark. And about 2 drops of solution X to the test tube. This is solution A. Solution A. And this solution is prepared from soap. So, add about 2 drops of solution X to the test tube with the solution A. This is solution A. And record the observations and the conclusions in the spaces provided. So, let's add together. So, the color changed. What is the color change? What's the color change? Say pink. So this is pink. Do you see the pink color? This is not purple. This is pink. So observation, you'll say that when 2 or 3 drops of solution X is added to the solution A, the color changed to pink. Now conclusion. Conclusion, you'll say that solution A is a base. Solution A is a base. Good. Let's do question 2. Add about 2 to 3 drops of solution X to the test tube with the solution B and record the observation and conclusions in the spaces provided. Solution B. Phenolphthalein indicator. Color change. Say pink. This is pink. Pinky color. So observations. What have you seen? When few drops of solution X is added to solution B, the color changed to pink. The color changed to pink. Conclusion, solution B is a base. So when a phenolphthalein indicator is added to a base, the color changes to a pink. A color change to a pink. Now, bases change to pink. All together? Let's do number 3. Add about 2 to 3 drops of solution X to the test tube with the solution C and record the observations and the conclusions in the spaces provided. C. Prepared from lemon. C. Prepared from lemon. Let's see what nature is a lemon. This is lemon. Shake. What's color change? Do you see any color change? No, the color remained. This shows that lemon juice is an acidic. Lemon juice is an acidic. So, phenolphthalein indicator, when added to an acid, the color remains to be colorless. So, the observation is the colorless color of solution C retained. The colorless color of solution C retained. That is colorless. This shows that solution C is an acid. Good. Four. Add about 2 to 3 drops of solution X to the test tube with solution D and record the observations and conclusions in the spaces provided. D. Solution D. Let's add. So solution D is prepared from sodium hydroxide. What is color change? The color changed pink. The color is pink. This shows that the solution D is unbased. So the observation, how are you going to put this observation? You'll say that when few drops of solution X is added to solution D, the color changes to pink. Conclusion, this is unbased. This is unbased. So in short, base is pink and acid is colorless. Acid is colorless. Now, let me take you to the next question. Name 2 basic scales that are necessary to carry out this practical. 1. Observation. We have observed color change. We have observed color change. This is pink color. So observations is a scientific scale. This is an acid. No color change. The color remains to be colorless. Phenolphthalein indicator, when added to acidic solution, the color remains to be colorless. Undash. Solution B is prepared from undash. So when this solution is added to solution B, I'm sure the color will change to be colorless. Therefore, this is an observation. So let's record this observation. When you put the points down to record, you record the observation. We call it recording. So there is observation using eyes to see the colors. You can also use nose to smell. It's also an observation. We can use ears to hear. You can hear some sounds. That's an observation. You can touch and feel the warmth or coldness. This shows that one is also an observation. Now, when you put the information down on a paper to record, to conclude, now I've recorded the observations. For example, I will say that, according to me, I will say that there is a recording to show what? Recording to record the observations, you see. And then number 2, after recording, we have conclusion. You can say conclusion. You conclude. Are we together? You can, from the first question that I've prepared for you, I'm telling you what are the observations. Then after observations, conclusion, conclude. Why did the color change? What made the color change? You see? So for me, the answer for that question about naming 2 scientific scales that can be used in such practical tasks, I can tell you we can write observation, you can write conclusion, and you can write recording, and you are correct. Question 3. You are provided with a block. You are provided with a block. You are provided with a block. Using a ruler, you are required to measure width in centimeters, length and height. Width, length and height in centimeters. Now, let's assume the length of this one is 4, height is 4, and the width is 4. So, width is 4, height is 4, and length is 4 centimeters. Centimeters. State the SI unit of length in this task. So the SI unit of length is meters. Determine the volume of the block in cubic centimeters. The volume of this block is length times width times height. But according to this cube, all signs are equal. So it will be length times length times length, which is 4 times 4 times 4, giving us 64 centimeters cubic. Giving us 64 centimeters cubic. Assume the mass of the block was 100 grams. Calculate the density of the block in SI units. Assume the mass of the block was 100 grams. The mass of this block is 100 grams. It's 100 what? Grams. Now, let's calculate the density of this block when the mass is 100 grams and the volume is 64 centimeters cubic. Density is equal to mass over volume. This is the density. This is the formula for calculating density. Mass over volume. What is the mass of this block? Say 100 grams. What is the volume of this block? 4 times 4 times 4. We got it as 64 centimeters cubic. Divide mass 100 grams all over 64 centimeter cubic. The answer is 1.5625. 1.625 grams per centimeters cubic. That's the density of this block in grams per centimeters cubic. Now, let's reread the question again. Calculate the density of the block in SI unit. So what we are supposed to do is you take 1.5625 grams per centimeters cubic and multiply it with 1000 to make it kilograms per meters cubic. You see how easy it is? So it is very easy. So when you want to change grams per centimeters cubic, to kilograms per meter cubic, what you need to do is to multiply that result with 1000 to get kilograms per meter cubic. So kilograms per meter cubic is the SI unit for density. Together? Yes. So assume the mass of the block was 100. Calculate the density of the block in SI units. So the answer we get should be expressed in, or must be expressed in kilograms per meter cubic. So the answer that we have gotten up here, that is 1.5625 grams per centimeter cubic. We multiply it with 1000 to get 1562.5 kilograms per meter cubic. Kilograms per meter cubic. This is the density of this block in its SI unit. So the SI unit of density we have said it is kilograms per meter cubic. Kilograms per meter cubic. Don't forget about this. Question 3. What is a cell? So we are now dealing with the cell. What is a cell? A cell is the basic structural, functional, and biological unit of all living organisms. It is often called the building block of life. Cells can exist as independent organisms like bacteria or as a part of multicellular organisms like plants and animals. So multicellular means made of many cells. Organisms made of many cells are called multicellular organisms. And organisms like bacteria, amoeba are made up of one cell, are referred to as unicellular organisms. So multicellular, many cells. Unicellular, one cell. In number 2, what are the differences between plants and animal cell? Differences between plants and animal cell. So for plants we have chloroplast, but in animals we have no chloroplast. That's the reason why animals cannot make their own food. Kisa na maana hazina chloroplast. Number 2, in a plant cell we have cell wall that gives the cell its shape. But in animal, we don't have cell wall. No cell wall in animal cell, but we have cell wall in a plant cell. Number 3, plant cells are regular in shape, but animal cells are irregular. Mwende from Joy Academy was using a light microscope to carry out a practical on plant cells. The eyepiece lens was adjusted to be x20 and the objective lens as x60. She was supposed to calculate the total magnifications of the cell. Help her to work out this using the knowledge you learned in science. The formula for calculating the total magnification of a cell is total magnification, that is total MAG, is equal to objective lens times eyepiece. According to our question, the eyepiece lens was adjusted to x20. You take it, you multiply with x60 to get 1200. How do we express this answer in x 1200? The answer is x 1200. That's the total magnification of the cell that Mwende saw using light microscope. I don't say that we are not bright, but according to our question, Mwende was not able to score that question. So that's the reason why you are helping her to calculate the total magnification of the cell. So Mwende, the answer of the cell that you saw using light microscope was 1200, and it should be x 1200. Name 2 organelles common in both plants and animals. What are common? What is common in both plants and animals? So what is common in both plants and animals? Number 1, we have nucleus. So nucleus is found in both plants and animals. Number 2, we have cell membrane. Cell membrane is found in both plants and animal cell. Number 3 is cytoplasm. Cytoplasm is found in both plants and animal cell. Number 4 is, kindly answer for me, what is number 4? What is common in both plants and animals? I've told you cell membrane, nucleus, and cytoplasm. Now, what is number 4? What is common in both plants and animals? I'm just checking the comments now. kindly send the comments. Haraka tafadhali. What is common in both the plants and animals? I'm waiting for the answers. In CBC, any answer is correct. Just send. I will correct. Send. I will correct. Then after sending, expect reply. Immediately, I will reply you as soon as possible. Check the comment section. When you comment, I will reply. Whether it is correct, I will tell you, good, it is correct. If it is not correct, then I will reply with the right answer. Send these comments. I'm waiting. Honestly, I'm just waiting to see the comments. I want to reply now. And to reply. Yes. Nimeona Marcus. Marcus, good. Nice. Another one from Honda of Tanzania. Welcome. Comments hapa. I want to see how many people are following, how many people are ready to do this Kenya Junior Secondary Assessment this year. Oh, nice. So like this video, share it with your friends, with your colleagues, with your classmates, with your candidates, mates, teachers. Share with your learners to see how to do practicals, Kenya Junior School Education Assessment. If you don't have these materials, just share and with your learners, and they will be able to do when it comes to an exam. So this work is an individual work, should not be done as in groups, should be done as an individual. So ni vyema mwalimu uweze kuambia wanafunzi wako, mwalimu waende watafute mwalimu YouTube, jina lake ni Teacher Ayubu Mwiti, na wataniona, na nitakuwa nimevaa kofia ya brown hivi, na nitafurahi sana kuona wanafunzi wanafuatilia hizi video. Tuonane tena kesho kwa majaliwa. See you. Bye.